by the way, just to reminder, your lecturer will finish the lecture tomorrow on Wednesday, okay, with the last part. And subsequently on Thursday, what is covered during lecture? Tutorial question 8 and 9. So my goal is I want to try and cover as much as possible before your lecturer goes through. Alright, now question number 3 is on monohybrid inheritance. Why? How do I know that? It's because I'm looking at one characteristic, which is the production of chlorophyll, okay? The color of the leaves, is it? Yeah. So anyway, you have one gene, okay, that is coding for the enzyme that produce the have you started? Okay, good. That produce the pigment, right? There are two alleles in the question. We have the allele CG and allele CW, right? CG codes for a functional enzyme which gives rise to green pigment. Okay, CW codes for a defective enzyme or a mutant enzyme. Okay, that gives rise to no pigment, right? So no pigment, what color is it? If there's no pigment, it will look white. Okay, so white is what you get usually when there's no pigment. Now they say that, right, you have a cross between two heterozygotes. Okay, so the two heterozygotes has to be CGCW cross with CGCW. Now, you're gonna see that a cross between two heterozygotes, right? What is the expected ratio? Expected. If you cross two heterozygotes, it's 3:1, but you don't get 3:1. You get pale green plants 2 is 2, dark green plants 1. So why is that so? Now if you write out the genetic cross, right, you're gonna see that this offspring will be this, and this, right? So let's try and put the phenotype with the genotype of the offspring. Now, which one do you think is dark green? 1, 2, 3, 4. Which one? Number 1, number 2, number 3, number 4. Which one is dark green like me? It has to be the first one that is dark green because I'm assuming that this functional enzyme is going to give rise to a darker pigment, more pigments, right? So what do you think is the heterozygote? What is the phenotype? It has to be pale green. Okay, and then there is a CW that is missing. How come there's no offspring that is like, that you see, why don't you have another phenotype? There's only two phenotypes. It is lethal. Lethal means the moment you have CW, the plant dies because no chlorophyll pigment to absorb light. Okay, so this is what we call a lethal phenotype. Because of that, the answer is D. Ken? Okay, so, yeah. Any questions? Okay, now the next question is, I think quite simple. It is what you have covered in lecture already. I just want to talk about some things to take note of if you were dreaming during lecture. So let me see. Now this question number 4 is a question on dihybrid inheritance. How do you know it's a dihybrid inheritance? Two characteristics, very good. So you have one gene, we call it gene A for flower color, and you have gene B for height of stem, okay? So that is your two gene loci, okay, for different characteristics. Now, if you look at the parents is homozygous white flowered long and homozygous pink and short. Now, homozygous, there is another way in which the question can phrase homozygous. What is another way they can phrase? Another term. They can say that it is pure breeding. Good. Pure breeding, or you can say pure line. Okay, that also means homozygous. Okay? Now this first parent, white long, crossed with pink short, you get an offspring that is white long. Okay? That's the F1. And when you solve the F1, you get 4 different phenotypes observed in F2. So there's 3 generations altogether, parent, F1, and F2. Okay, now let's try and figure out what is the parents first. Now what do you think is the parents? Or what do you think, okay, let's figure out the alleles first. Now what do you think is the dominant allele? Big A. Okay, the dominant allele, big A, for which flower color? White. White. How do you know it's white and not pink? F1. Very good. You look at the F1 generation, right? If you cross 2 parents that look different, but you only get this offspring phenotype, it means that it has to be the dominant trait. Okay, so white is the dominant allele, big A, pink is the recessive allele, small a. Now for height of stem, big B and small b. Big B is, it has to be long stem, right? And what is it? Short. Short stem is small b. Okay, so that's how you figure out the dominant and the recessive allele. Now, so given that, can you figure out what is the parental genotype? Let's say you use these alphabets A and B. Parent, homozygous, white, long. Big A, big A, big B, big B. Okay, the other parent, small a, small b, small b. Now, you don't even need to do the genetic cross, you should already know what is the F1 genotype. Both gene locus is a heterozygote. Okay, now then you see the F2, what ratio are you expecting to get for F2? Based on the ratios you have memorized. 9:3:1, because you cross two double heterozygotes. Now the next question that students will ask me is this question: do I have to start with the parental generation in my genetic diagram? Should I just do F1 and F2, or should I go all the way from the parent to the F1 to the F2? Which one? That's a very valid question. You should do all the way, starting from parent. How do you know? Okay, you look at the number of marks. Okay, generally speaking, a dihybrid question is usually 4 marks. But in this question, it's 6 marks. So it means that you have to show me something more starting with a parental generation. That's how you explain. So I'm going to show you the answers. It's not difficult. Okay, I think you should know how to do this. So this is what you should get. Okay, I'm going to show you up to F1 first. Can you just mark your own answer? So the F1 is up to here, basically. Okay, can you please note where are the marking points? Okay, I repeat, a lot of students, they don't know what is the marking points. Make sure you know. So 1 mark is for the parental genotype. The phenotype is given already. So the genotype is 1 mark. 1 mark is for the gametes that are circled. 1 mark is for the F1 genotype and phenotype. Yes? No need. Question, do you need to write that the parents is 2N and then the gametes is N only? Okay, another question that's commonly asked, do you need to draw the arrow showing meiosis? No need. What else is asked? Do you need to write the cross like this here? That's another question. This one, this cross, do you need to write? Yes, because you're doing a cross, a fertilization, right? You have to write the cross. So even gametes, you have to circle it. Now what is a common mistake by the weaker students? Usually they will draw the gametes like this. Okay, please note that you cannot draw gametes like this. Why? Because based on Mendel's law of segregation, you should know that when you have a pair of alleles, okay, these are 2 genes, not just 1 gene. A pair of alleles, each gamete should receive 1 allele of the allelic pair. Alright, so you should have one big A and 1 big B. Alright, so that's the mistakes normally. Do you need to write the key, the symbols, yes or no? For this question, you have to write the symbols even though the alphabets are given, because you want to show the marker that you know which is the dominant allele. Okay, so that will be good if you give. Can? Have you taken a photo? Okay, then I'm going to scroll down and show you F2. Can? Okay, now F2 generation, so I'm going to zoom out a bit. So if you were to, starting from here, okay, the F1 generation was self-pollinated. Now in plants, it's possible to do selfing. You cannot do it in humans. How come plants can do selfing? Self-pollination. Yes, self-pollination. Yes, okay, for flowers you have the anther, which have the pollen grains, and you have the stigma for the female. So you can just transfer the pollen grain to the stigma, so it's self-pollination for the plant. But humans, you can't do it, okay? So anyway, so if you're going to self the F1, these are the 4 types of gametes that you get. Now over here, this is usually the weak students, this is the part that they cannot do. So how do you know there's 4 types of gametes? Who says so? Which law tells you that there should be 4 types of gametes formed? Law of independent assortment and segregation. Right, because the homologous chromosome, we are saying that these 2 genes are on different chromosomes. So when they arrange, they can arrange in different ways to give you these 4 different combinations. Alright, so can you look at the mark scheme? The mark scheme is the F1 gametes are 1 mark. Must it be circled? Yes, as long as it's gametes, it needs to be circled. Can you check the Punnett square? Oh yeah, there's a typo here. Okay, so there's a typo. Can you please check? Alright, so question, is it compulsory to draw Punnett square for this question? Just now, do you notice the Punnett square for your parents? No, that one don't need. But for this one, you need to. Why? Because it's more complicated, right? You need to show that, oh, I fuse this with this gamete, I get that. Okay, so you need to draw a Punnett square. Another question, do I need to draw this gender sign, this symbol? By the way, you know, right? The male is the arrow. That makes sense, huh? The female is the plus, okay? So that's the male and female. Now for this case, for plants, right? Okay, now it's not specified in the question, right? So when you assign the gender symbol, it's random. But there are some questions that you're gonna see, okay, I'm crossing a female this with a male that. Then you have to do the gametes correctly. You get what I mean? So it depends on the question. So this question, the gender symbol is random. Okay? Now another question, this one very leceh, you know, wow, I need to write white long, so complicated. Can I don't write? No. You have to write because you're given marks for this. Okay, so if I scroll down, you see the mark scheme, right? You're given 1 mark. I mean quite pathetic, 1 mark to show both the F2 genotype and phenotype. So you have to write. Sometimes students ask me, like in exam, I desperate already, got only 1 minute. Can I do short form? I write like this, white long, white short. I mean, can, but try not to unless you really got no time, okay? Some of you, if you write WL, WS, make sure you have a key at the side to tell the examiner what WL and WS mean. Okay, if you are really desperate, no time, okay? Alright, so can you please note all the marks? Alright, so 1 mark for the gametes, 1 mark for the Punnett square which shows genotype and phenotype, 1 mark for the phenotypic ratio at the end. Any question? Now the last thing is, if this question comes out and they give you the observed number, so in this case, right, what you get at the bottom here, this is the expected ratio. Expected. Right, sometimes in the question they give you observed numbers, they tell you there's 90 plants, and then there's 31 plants, and then there's 32 plants, and then there's what? 15, something like that. You have to actually relate it to the observed numbers if it is present in the question. I repeat, if the question gives you observed numbers, you have to correlate your expected to the observed. But in this case, there's no numbers given in the question, so you do not need to correlate. Can? Okay, now based on this, have you taken a photo? Taken. Okay, so can I move on to the next part? I think this part should be no issue. It's similar to your tutorials. Your lecture. Now the next part is this one. They say that in another cross, I have white long plant with unknown genotype crossed with pink short stem plant. Right? And I have all these offspring formed. Now what is the point of them giving you numbers? These are the observed numbers, like I mentioned just now. Why do they give you these numbers? For what? Good, they want you to show ratio. Okay, so the ratio here is 1, 1, 1, 1. Okay, now let's try and figure out the parental generation. Right, the pink short plant, that one very simple. It has to be small a, small b, small b. That one no issue, right? Pink short. Now the white long, it has is unknown. So this is the possibility. But based on what you have memorized already, 1, 1, 1 ratio, how do you get that? It has to be a heterozygote and both gene locus. Okay, so that's what you get. Now, in terms of the explanation, okay, they say, why is it that the results of this cross differs from the cross described in A? Now, which cross are they referring to? The cross described in A. Are they talking about the parental cross or are they talking about the F1 cross? F1? No, they are talking about the parental cross. Okay? Why is it the parental cross? Because the parental cross is the same phenotype as this. Okay, parental cross, you're also crossing white long with pink short. So the parent that is in part A is actually homozygous dominant parent, also crossing with this plant. Now, what do you think is expected in your answer for 4 marks? First mark, what would you mention?
You say about the genotype.
Yes, okay, 1 mark. Actually, 1 mark. This is actually a technique. Do you realize when you have an unknown parent cross with a homozygous recessive, what is it called? You learn about this?
Oh, test cross.
It's actually a test cross. So the idea that it's a test cross is 1 mark. Second mark, you have to talk about the parents here. The parents here has to be a heterozygous at both gene locus. Why? Because when it crosses with this small a, small b parent, you're going to get a 1-1-1-1 ratio. So that is your third mark. And then the last mark, what do you think is the last mark? Okay, how do you explain that I am sure I'm gonna get 1-1-1 ratio if I cross this with that? Who says that? Who tells me that I'm gonna get 1-1-1-1? Who? Mendel. Mendel is the guy who tells you that, right? So based on Mendel's law of independent assortment. So let's write out the answer. First mark, this is a test cross. Second mark, you're gonna tell me that the unknown parent, the white flower, long stem plant, okay, that is unknown. Okay, basically the parent is heterozygous at both gene locus. Okay, so he is AABB instead of the other parent AABB. Okay, instead of being homozygous dominant. Right, because he is having this, it is having this heterozygous gene at both gene locus. What happens is, according to Mendel's law, I'm going to form 4 different gametes. Gives 4 different types of gametes. Which is, if you want, you can draw out, okay, this, right? Instead of if it's the other parent, it is just this. Right, this is due to the law of independent assortment and segregation as the 2 genes are on different chromosomes. And the last mark, therefore, when you cross this parent, when crossed with a homozygous recessive plant, okay, you get a offspring ratio of 1-1-1-1. Okay, so can I zoom out? Okay, so sorry, we have to wipe down already. I forgot there's another class. Okay, so can you go and take the wipe down material?